Tēnā koutou katoa, ko Vivian Handaho, no mai, haere mai ki te Aotearoa Food Safety Science and Research Centre. Ka nui te mihi nui kia koutou katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, I'd like to thank you all for joining us today to join our regular monthly focus on food safety science seminar, the Spotlight Seminar. Today we're very privileged to have Dr. Anu van Vliet, who's joining us from the UK at sort of almost 11 p.m. So we're really grateful for him to join us. And some of you will know Dr. Anu van Vliet is the Senior Lecturer in Microbiology at the School of Veterinary Medicine at the University of Surrey in the UK. His research is focused on zoonotic bacterial pathogens such as Campylobacter, Listeria, E. coli, and Salmonella. And while he did train as an experimental scientist, Anu moved into the area of genomics and bioinformatics with the aim of understanding, using genome sequencing and analysis technologies to better understand bacterial virulence mechanisms, the immune escape, phage defense, and antimicrobial resistance. Anu has been an active member of the Centre's International Science Advisory Panel since the Centre was first established in 2016, and very grateful for him for being a night owl. And I'll hand over to Anu now to give his talk on genome sequencing and update and some of the pitfalls. So Anu, thank you very much.
Thanks very much, Vivian. Thanks for the invitation and the chance to talk to you all. And today we're going to talk about food safety and whole genome sequencing. And I hope you can see the screen. Well, that's okay.
Yeah.
Unfortunately, I was hoping to be able to do a Zoom poll here, but as I'm not the organizer and it's on the messy one, unfortunately I couldn't do that. So I may ask you at some point your question. You can unmute if you want to answer it. If we go throw it into the group. But first, let's talk about a bit of genome sequencing. And those who had read the abstract may have seen something that I said, the disruptive invention or disruptive technology. And genome sequencing can be considered one of those. Now, is genome sequencing as good as sliced bread? Or is it like this little clip from the, I'm old, I'm 50 plus, so just from Genesis, Land of Confusion video clip, where you can either ask for the nurse or for the nuke. This was from Ronald Reagan. Pressing on. But what is a disruptive technology? It's a technology that suddenly changes everything. And for instance, you can see here one of those graphs from Wikipedia. You got a low quality reason, everything is progressing in a certain way. And then suddenly this new technology changes everything, changes the playing field. It also changes the expectations of what is possible. And so these kind of inventions can be very much a game changer. Now, I wonder whether anybody from their own life, we all use disruptive inventions or disruptive technology. Feel free to unmute yourself if you want to name one that you're using yourself, that you recognize as such. So does anybody know like something that uses
Uber?
Uber? Yeah, I'm not sure whether that's a technology, but I can see the idea with the booking of the taxi making so much easier.
Cell phones.
iPhone.
Cell phone.
Yeah, iPhone is absolutely one of the disruptive inventions. Anybody know another one? Well, I'll show you a few. The iPhone was already mentioned, but one of the earlier ones, for instance, was the 4T, when it suddenly was produced at mass, and people could just buy a car and drive it very easily. The compact disc, where before that you copied music, and it was a lot of loss of quality. We've done that a lot when I was young. The compact disc suddenly meant you could copy it without losing anything. But that also led to Napster, the illegal file sharing, and nowadays we got the more legal version with things like Spotify and so on. It's completely changed the music industry, completely changed on how we consume music. And I use Spotify and everything a lot. I don't have a car and I don't have an iPhone, but it has really changed things completely around. And genome sequencing, and within biology, PCR, where things like disruptive inventions, they completely changed the playing field. And if I wanted to just quit the presentation now, I could say, why is genome sequencing the best thing ever invented? Well, the COVID pandemic has shown how genome sequencing can really help us. We wouldn't know about any of the variants that are doing the rounds if we wouldn't have had genome sequencing. This is from Gizade, this is from one of the experts on COVID. And this shows how much the Delta variant is suddenly taking over in the world. This is a phylogenetic tree of hundreds of thousands of COVID genome sequences of SARS-CoV-2. And without this genome sequencing, we wouldn't have been able to manage the pandemic as we have been and find out where infections came from so quickly. So it is really we've been lucky that we had it available to us. But there's a lot of details because genome sequencing is just a technology, but there is so much information variability. It actually started in the 1990s, but now everybody's using it. It used to be extremely expensive. And we did one Campylobacter genome sequence. I was involved in the first one, and that cost us more than £500,000. So that's more than $1 million New Zealand dollars. And we got one genome sequence for it. We didn't know anything more than we knew before, and we had a Nature paper. So we could call it a very expensive gold open access by now. But nowadays, it's actually quite relatively cheap. But how many people actually know about the knowledge on details? And this is why I want to do a Zoom poll to see how many people who are listening to are actually doing genome sequencing. Because for a lot of people, it's just a couple of black boxes. They give their sample, they get something back, but do they know anything about what happened in between? And once they get their data back, again, they quite often hand over their data to bioinformaticians who do the data analysis. And again, there's a black box that they really don't know the details of what's being done. And if you don't know how something works, at least the principles, how can you troubleshoot? How do you actually know whether you have good data or bad data? And that is a big problem now with genome sequencing. We had it with PCR as well. With PCR, at some point, everybody started using it. Also, people using it completely wrong and drawing the wrong conclusions. And that's the downside of disruptive technology. Everybody gets their hands on it. And there is also, a lot of people are not used to suddenly these changes in the playing field. And this is one from a professor of bioinformatics, Mick Watson, who can be very provocative. And he basically once said that most bioinformaticians are bad scientists and most biologists are bad bioinformaticians, which makes me, as a biologist who became a bioinformatician, probably a super bad scientist. But he also said very truly that bioinformatics has really failed to enable the non-expert user, and I'll come back to that. There's also this thing, what I do sometimes with people, they think almost like it's magic. And then there's Arthur C. Clarke, a science fiction writer from 2001 Space Odyssey, for instance, who has a couple of laws. And one of his laws is any sufficiently advanced technology is indistinguishable from magic. And the counterpoint is always make that any technology distinguishable from magic is insufficiently advanced. But basically what you have is you have biologists, and you have informaticians, and quite often there's quite a big difference in how they speak with each other and whether they can understand each other. And so we really need people to be between these two and help translate. And this is very important in any multidisciplinary team. The other thing with sequencing technologies is that, of course, it's an open field. And I've adapted this well-known XKCD one about standards. This is how sequencing technologies proliferate. We first have 14 competing sequence technologies, and that's ridiculous. We really should all decide to use one so we can all do the same thing and compare data. And then, of course, after we decide on that, nobody wants to give in, and then soon we have 15 competing technologies. We're scientists, we're academics, we don't make it easy. Now, briefly, what are the sequencing technologies that are really leading? So there's really 3 technologies that are still standing. The best known one, that probably is in most laboratories that do sequencing, is Illumina sequencing. Started out as Solexa, was bought up, is now called Illumina. And basically what happens is you have here pieces of DNA that are immobilized somewhere. They're single-stranded, and the DNA polymerase start adding fluorophores, so labeled, fluorescently labeled nucleotides. And each time one is added, it gives either a flash or not in a certain color, and that flash is recorded, and so then they know that that nucleotide has been added. And so a lot of images are taken, and that is then translated into the sequences that you get. It's because it's quite time consuming and very data heavy, you can only get short reads, usually somewhere between 100 to 300 nucleotides. And if you think about that, for instance, if you compare it with Listeria, that's only 0.01% of a Listeria genome. So you need a lot of reads to actually make up a genome of a bacterium even. It's very high quality, but low error rate, lots of data output nowadays. And basically what it is, it's an expensive machine, and it's not portable, but you can make very low cost per sample. So you can do a lot of samples for the money you have. And so it's used for genome sequencing, metagenomics, RNA sequencing, all these kind of technologies. You get packed biosequencing, which is sort of like losing and moving out slowly. That's sequencing as well with fluorophores, but there it's not actually measuring the flash with a fluorescent light, but they are actually sort of like having a camera following the polymerase. And each time a nucleotide is built in, it takes a certain amount of time to build it in. And that differs per nucleotide. So an A, C, G, or T will each have a different time span that it takes for them to be built in. That time span is measured, and so then it's translated on which one was built in. It's actually get very long reads because it just keeps on racing around the DNA molecule. But it has quite a high error rate, so it's not very good quality sequence. So you need to sequence a lot still to get decent sequencing. It's an expensive machine, again, not portable, and the cost per sample is a lot higher. So not a lot of people are doing this. And then there's the one that probably most people have heard of, that's the Oxford nanopore sequencing. That's the hot new thing on the market already for a few years. And it basically pulls a bit of DNA through a pore, and it measures the disruption of the electrical pulse. And then again, for each nucleotide, that disruption is different. So it will tell you whether it's an A, C, G, or T by the length of and the type of disruption. Again, high error rate, low data output. And this is one where the machine is very cheap and portable, but the cost per sample is high. So this is a bit like the Gillette shaving or the inkjet printers. You basically get the handle or the printer for free, but you pay through the nose for the consumables. But this is the hot new thing, and it's getting better and better. And the nice thing is that it's really portable because it's like a big USB stick, the first one, the MinION, which is the best known one. Then you got the GridION and the Promethean that are just a lot of these MinIONs together. But you also have even one that can run for the Snitchen that runs from the mobile phone. So it's powered by a mobile phone, and the mobile phone actually does the recording of the data. So this can be done anywhere. So anywhere in the wilderness, on the space station, you can sequence everywhere. As I said, just making the sequencing doesn't get you anything because you need to analyze the data. Now, I've shown here a simple picture for genome assembly. It's basically you have your genome, you break it up in lots of pieces, these are sequenced, and then you try to put it back together like a puzzle. You got what we call the complete genomes, which are a very few of the genomes, and most of them are, everything is probably there in the puzzle, but we don't know how good quality it is. There's a bit of rubbish in there, so the quality may be a bit lower, but those are the ones that are easy to produce. And as I say, analyzing genome data is a bit of a black box for most people. Now, there's usually this thing like if you want something, you can either have it easy or you have it good. So it's either quality or it is easy. The good quality programs are not easy to use. So then you get this kind of command line that people will shut down straight away, where nobody has a clue about what's written here on this picture. Let me just put the pointer option to laser pointer. And the ones which are easy don't give good quality outputs usually, or not decent at most. And the problem is that the programs that people use, and I like this cartoon very much here. So how are you with Excel? Well, I hate it. Oh, you're an experienced user then. And what the joke is in computer people is that most of the problems are caused by PEPCAC. The problem exists between the keyboard and the chair. That means a user. So we make our own problems. Okay. That's for a bit of background information, a bit of insight. So now some examples. So from my own practice, what can you do with genome sequencing? AMR is one of the hottest subjects after COVID nowadays still. Antimicrobial resistance is a big problem. We're running out of antibiotics. And so something that was proposed to deal with that was antimicrobial stewardship. So it's a set of coordinated strategies to improve the use of antimicrobial medication, to make sure that we still get good patient health outcomes, but we don't get antibiotic resistance, and we lower the cost. And the concept is that antibiotic resistance is expensive for bacteria. So once bacteria become antibiotic resistant, they have disadvantages. But if they're growing together with other bacteria with the antibiotic, they are actually ahead because they are resistant and the others can't grow. And the idea would be that the absence of antibiotic would select against the resistant bacteria, and that's shown here. You got two non-resistant bacteria, two resistant bacteria. If you're growing without antibiotic, then the antibiotic resistance becomes a disadvantage. So they can't grow very fast, whereas the ones that don't have the resistance can outgrow and outcompete the resistance bacteria. So by using less antibiotics, you should be able to get rid of the antibiotic resistance again, or at least lower it. So the question we wanted to say is, can we actually see that concept in action? And for that we used Campylobacter jejuni and coli genome sequences from the UK and the USA. The good thing is that both these countries have surveillance programs, so they're sequencing a lot of Campylobacters and put them in public databases. So you can download them and use them. So I had just over 32,000 Campylobacter jejuni and almost 9,000 Campylobacter coli genomes. For those, I had the metadata available, so the year and the country. I determined their antibiotic resistance gene content, so what antibiotics they should be resistant against, and focused on the four main categories of antibiotics. And what you then got is here a bit of an overview. You see here the number of samples for Campylobacter coli and for Campylobacter jejuni. And you can see from 2015 onwards is the US is really starting to sequence a lot of samples. So in 2018, already more than 2,000 a year. UK keeps on steady around 500. And with Campylobacter jejuni, the UK is a little bit up and down, depending on what people submit. And again, the US is sequencing up to 4,000 already per year. So a lot of samples. And here I had a quick look at where they came from. Did they come from farm animals, from poultry, or from humans? In the UK, a lot of them came from humans. In the US, it was a bit more varied. That's the way they're sampling, because they're sampling at the farm, they're sampling at supermarkets, and they're taking clinical samples. And then here you see over the years the percentage of resistant strains, genomes in for the different antibiotics. So for the aminoglycosides, for the Campylobacter coli, and for Campylobacter jejuni, for the macrolides, for the quinolones, and for the tetracyclines. And one of the things that I hope you can see from it is that it's actually quite stable. There is a little bit of variation from year to year, but from the number of genomes, we can at least draw quite a few conclusions. And what we can see really is that this not using antibiotics doesn't really do anything anymore. So the number of resistant bacteria stays quite the same. You can see it for tetracycline especially, no change. For quinolones, not a real change. Luckily for the macrolides, it's still low for Campylobacter jejuni because this is one of the clinically used antibiotics. And aminoglycosides are completely useless against Campylobacter coli, but again, it stays stable. And there's not real big differences. It's in the level more between the UK and the US, but there's not a real difference, so it's not really going down. So from this data analysis with genome sequences, it looks like the concept of antimicrobial stewardship is not really working. So we can already say that we can't use tetracyclines, we can't use aminoglycosides. We need to be very careful with quinolones in humans, and macrolides also we need to be very careful with. So maybe we're already too late. So that's the doom message done. But there's also positives, because why can we do this? So that's the power of data sharing. And this is a second example. This is the NCBI pathogens database. So this is where all those data are uploaded. And especially the US is uploading a lot of data. And you can see, for instance, this is a screenshot from a few days ago. You can see that they uploaded almost 500 salmonellas in one day. 59 E. coli, 25 Campylobacters. They got a whole list of pathogens that they actually are uploading data from. And if they're already combined with the data from small local studies, you get enormous amounts of powerful data for these kind of studies. And you can see a listeria, very important one for New Zealand food safety. You can see there's almost 45,000 genome sequences already. And I've had a play with 38,000. And this is just a little bit of where they come from. That's the most important one. You see the majority of those are American, the yellow ones, North America, so that's Canada, especially US. And the red ones are Europe. Africa is the big one that is missing. There is not a lot of surveillance done in America, in Africa, and that is one of the problems that we really need to deal with at some point. And then once you start analyzing these kind of data, as you can imagine, this is one of them where you really hammer yourself. You get a lot of data. I'm not going to explain what's on this one. I just want to show a very data-rich slide. And you can basically start looking for a lot of different things, like what genes are overrepresented in the clinical isolates, what genes are underrepresented. Can we find anything that could explain why some listerias are very successful? And that's an ongoing situation. But the problem that we find so far is that any of the known markers that people are using in studies are actually not informative. And so we still don't really understand whether there are listerias that we should not worry about, or whether all listerias are bad. And then finally, some opportunities. This is something that people need to consider. What good are these data if people can't analyze them? And you know, the PEPCAC and all these kind of pictures that I showed are quite often quite negative about users. So what good are open data and repositories if nobody can use the information? And also, there's Maslow's quote, if all you have is a hammer, everything looks like a nail. So if you don't know how to analyze things, just sharing data is not enough. You actually need to have an infrastructure that people can use and teach them how to use it. And preferably it should be able to be done on your laptop or your desktop computer and not just on a big power cluster. And so I've set up actually genomics training, and that's based on the Lao Tzu one, you know, the Taoism founder. Give a person a fish and you feed him for a day and teach him how to fish. You feed him for a lifetime. So I've done the bioinformatics equivalent of fishing education. My dirty mind is a joy forever thing. I had to laugh about this website about tailortackle.com, learn how to fish. And basically what it is people get a virtual Linux computer with all the tools installed, and they're all open access, so they're free to use. They get example data, and they get something that works. They get a series of instruction videos, which is especially important nowadays with, especially with online education and people being everywhere in the world. So you can't just ask me to come over to teach you. And also this computer can later on be used for people to try their own things. And then they get me talking in like 20 to 40-minute videos, several of them, about the different aspects of how to make phylogenetic trees, how to do antimicrobial resistance analysis, and all these kind of things, and includes my bad drawing on a tablet. But it basically means that everybody can do it at their own speed and at their own computer. And I've got here a few of these kind of comments that people sent me when I asked for them. And the most important one from these testimonials were things like, they felt that bioinformatic expertise was sequestered with people. So people that knew how to do it wouldn't tell other people how to do it, so they still kept their importance. They felt it was a very mystical thing, and so this demystified it. It's worldwide accessible because as long as you have a computer, you can do it. And it ensures that the expertise is shared. And because I set it up myself, the program should work like they're supposed to. Otherwise, I wouldn't share it. And actually, people started doing their own analysis of sequence data, despite not having a lot of experience with it. So it actually does work. And this is available for people if they want to. They just have to contact me. Now, finally, I promised some headaches. And that's only the one from all my slides, probably, in the information. But this is one study, one slide only, and it's basically lots of people work with reference strains. So they work with their Campylobacter, their Listeria, and they all have the same strain that they're supposedly using. Well, within the UK, we actually asked, is that actually true? Are we all using the same strain? And so they collected strains from all over the country from people's collections. They sequenced them and compared them for Campylobacter jejuni, and it was one big mess. Nobody's working with the same strain. Everybody has a different one in their freezer. So officially, most of the results aren't even comparable with each other. And this is one of those questions, once you ask the question and you get an answer, you need to do something with it. That's the next step that we still need to take. But this is a big headache that will come up with lots of people working with reference strains. So to finish up, what can we do with this? Briefly, currently we go into factories, we go to patients, we swab them, and we grow them, and we sequence a single isolate, and we get something really nice, but it's slow. It underestimates the diversity because it depends on which of these colonies you pick. If you can't sequence it, you're not going to find it, and so you may have very low sensitivity, and this is the gold standard. But maybe we could just start doing these things in factories with, for instance, the Oxford Nanopore one, where we could swap there and start sequencing everything is there in the sample. So we don't need the culture. We have rapid testing possibilities. And if we combine it with the standard surveillance, we actually may understand a lot more of what's happening in the food chain, in factories, and how person-to-person transmission is happening. So there are still an enormous amount of possibilities that can happen. And so to conclude, it's highly informative, and industry and regulators, government will need to deal with this. It's very important they know how to do it as well. It's extremely powerful for retrospective analysis, for outbreaks, but that also means that data sharing agreements, especially with industry, this is of course very sensitive. They need to be very carefully drafted of what information can be shared and what cannot. We need to work on prospective applications and also especially how we translate this into risk assessments. So we get sequence data. How can we say what the risk is that we do? And we actually have a position that we hope to fill soon with somebody working with our food standard agency to start looking at that. And the COVID pandemic is actually telling us what we need to change and how we need to deal with data. So have a look out for how they're doing it and learn from their wins and their losses. With that, I want to finish. Lots of people to thank. Here are just a few, including the people from New Zealand that I'm very grateful to be able to work with. And thank you for your attention.
Thank you, Arno. That's really useful, really helpful. So please send some questions or fire away some questions that you might have. One I have was actually around what you said about the Listeria monocytogenes, and you said that some of the markers that people are tracking are not that useful or not that informative.
Mm-hmm.
And so, sort of what, I mean, what, do you want to just, could you explain a bit more about that? So, I mean, that's listeria is a big, not a big problem, but a concern here, as you pointed out. So I think it was, which slide it is, it's when you did the metadata on the listeria genome assemblies.
Well, what it means is that a lot of people do studies which are relatively small scale. We've done one in New Zealand as well, where I had a couple of hundred genomes, 200 genomes, and we got a bigger database which is about 1,000 genomes for New Zealand. But lots of people do a local study, and they then do the marker genes. So they look for the stress islands, the pathogenicity islands, the plasmids, the biocide resistance genes, all these kind of things. But they actually show a very strong, I'll go back to the picture. Let me just share it again. So basically what you see here is that the tree is quite structured. And whether a gene is present or absent actually depends on the position in the tree, this clonal complex, as it says here. So if you have a small scale study, the only thing it means is that you get, from doing those marker genes, you get the same information as you get from the multi-local sequence typing. It just tells you about the diversity of your local population. It doesn't tell you why some of them may cause clinical infections and others do not.
Okay.
So it doesn't, doing all these analysis on a small population of listeria strains from your own area doesn't really add much to the multi-local sequence typing. With maybe the only one where I think it's really useful is probably the biocide resistance, because that tells you something about the cleaning regimens that you can use.
Okay.
And that's what I tried to say with this. And there's so much data that it's very difficult to show, but it is really, the problem is that none of those marker genes actually tells us something about why some listerias, for instance, the yellow ones here are the clinical isolates. You can see that CC321 has very few clinical isolates, whereas CC9 has a lot more of them. But why is CC9 more capable of causing human infections than CC321? That isn't answered by those markers.
Great. Well, I mean, I really like, and so this course that you run, you have that people from industry engage in that and start doing that, because that's one of the big challenges, is the information that you gain from the work.
Well, at the moment, it's only academics who have used it. It is on offer. I've already discussed it with the Food Safety Center here. It just needs people to take up on it, and everybody's very busy, of course. But it is something that is allowed. I mean, it's something that I just made to make sure that people can benefit. I made it for our own PhD students first, because they needed to work with those data and actually needed to defend it when they did their PhD viva. And then other people said, well, but I want to be able to do it as well. So at first I did it in person, and that was like 2.5 days of really intensive, absolutely headache-inducing kind of work. And so when the whole pandemic started, I said, well, actually, this is perfect for doing this with videos where people can do it at their own speed. If they get stuck, they can always contact me and ask questions, but they can do it whenever they feel ready for it. And work with data that I provide first, and then try it with their own data. So yeah, it's really a learning thing.
Yeah, it looks great. Any other questions from people? Everyone's gone quiet. I had one other question, is, you made the point about the reference strains being different. Do you think that's likely that problem in other countries?
Absolutely. It basically means that, it's a worldwide one. We just did it in the UK. I think some people have done it for Pseudomonas as well and found similar problems, that this PA01 strain that everybody is using, there are very many different variants around. Campylobacter is probably worse in this because it's genetically very variable and unstable. But it means actually that, even your E. coli that you're using for cloning may not be what you think it was at the time. And it's similar to many of the tissue culture cells that are in collections, which are contaminated with HeLa cells.
Yeah.
And basically people thought they were doing experiments with a heart cell line, and it turned out they were trying this on HeLa cells. And that's the problem. Yeah. But if you don't look for it, you're not going to find it.
No, I don't know what work's been done here. I'm sure. Yes, I'm sure it's an area that people are worried about. Just a question from Malik in Melbourne. He's saying that SMEs are not really in a position to afford expertise in whole genome sequencing technology. So, how can it help the food industry manage food safety?
This is I think they can't have somebody specific for it. They could train, of course, staff in it, but depends on how much staff turnover there is, whether you have to retrain people regularly. What I would say is that SMEs could actually band together, and rather than having every SME doing it themselves, is actually work together on these kind of things, so they don't have to bear the full cost of it. Most people don't have a sequencer. We now have a sequencer at a university, and we're not using it. We just send our samples off elsewhere. Now, I can understand for industry, again, that may be a little bit sensitive, that they don't want to send their samples away, but I'm sure they can find ways of making it all confidential and not having any data, because the sequencing itself doesn't contain any data, it's just reads. It's very difficult to abuse that if you don't have any of the metadata. And it's the metadata where the power is really in. So I would say that SMEs can actually do it. They may have to work together, maybe do it together with ones which are not competing in the same sector. So again, you don't have this problem as information leaking out. But I'm doing all these things myself. It is not that difficult once you know how to. Yeah.
I mean, I think some of the challenge for industry is the bioinformatics part, isn't it, and reading the data. And that's where the centre tries to play a role. And we have a number of people in our centre that can help and making sure that the information is being used. I think we've been quite useful for looking at biocide resistance and looking at treatments in plants. But again, probably small scale studies because we haven't had the big data sets. Any other questions from everyone? People are very quiet. I think you've given obviously such a good talk. I know you've explained a lot. I liked your first reference. So you were involved in one of the first use of whole genome sequencing in the 90s, were you? In the Nature paper.
Yeah, that's my only Nature paper. So that's the way to do it. But it's the first one. And it started, I was still cloning things and did 3 months sequencing a 3 kilobase fragment, and then genome sequencing started. And within a few weeks, we had more information than I generated in a whole year.
Yeah, fantastic.
It's also very demotivating from time to time. You have to move with the times, and of course, the opportunities are enormous. But it's been a rough ride from time to time, but a really interesting one. And one of the things for PIs, people who don't have the chance to go into the lab anymore. This is something you can do at your desk. You can stop and start it easily. So this is my way of doing science while still having all my admin duties.
Exactly. No, that's right. Oh, that sounds good. Well, look, I think I don't have any more questions. I've had a few people saying great presentation. So yes, I think it was a very good presentation. Thanks, Arno. And I think we had a record number, actually over 66 on the call. At one point, people are starting to drop off as they always do on a Friday, but you know how it is. So I really appreciate your talk today, and we've got another talk at the end of July, which will be the 30th. We're going to talk about hopefully with Ag Research, talk about some of the projects on the blue LED, use of blue LED light to sterilize plants. So we're going to have a discussion on that. So thank you very much again, Arno, and you can go to bed now. Or not. Anyway, well, look, thanks very much, everyone, for calling in, and we'll talk to you again on the 30th of July. Thanks, Arno.
Excellent. Thank you all for your attention.